Welcome to Microbiology classroom. Today I want to describe about the diversity of the fungi, various groups and classes of fungi. Eukaryotic microbial diversity, fungal diversity. So diversity of fungi. The fungi are large diverse group of eukaryotic microorganisms. Fungi are a large diverse widespread group of microorganisms. So far 1,20,000 fungi are described and discovered. Approximately 1,20,000 species of fungi have been described and discovered. There are estimates that more than 1.5 million fungi are there on the earth. So there are estimates that more than 1.5 million fungi are there on earth. In 1991, one research paper estimated that there are more than 1.5 million fungi on the earth. But recent analysis of environmental DNA samples, metagenomic study analysis of environmental DNA samples from a soil community reveals that a high rate of new species accumulation at that site, and these data supporting that there is an estimate of 3.5 to 5.1 million fungal species in soil. Fungi, phylogenetically, fungi share a more recent common ancestor with animals than with any other group of eukaryotic organisms. Phylogenetically, fungi and animal cells are sister groups. Fungi and animals are thought to have diverged approximately 1.5 billion years ago. The earliest fungal lineage is thought to be the Chytridiomycetes, an unusual group in which cells produce flagellated spores, juice spores. The lack of flagella in most fungi indicate that motility is a characteristic that has been lost at various times in different fungal lineages. The true fungi are divided into four classes by Saccardo in 1886. Saccardo in 1886 divided true fungi into four classes: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. This four-class system of fungal classification dominated for a long time and was accepted by many mycologists. Later, Sparrow in 1958 recognized four classes within the aquatic Phycomycetes. These classes are Chytridiomycetes, Hypochytridiomycetes, Oomycetes and Plasmodiophoromycetes. The two other subclasses, Zygomycetes and Trichomycetes, these are also given the rank of the class. So there are 9 different classes in fungi: Chytridiomycetes, Hypochytridiomycetes, Plasmodiophoromycetes, Oomycetes, Zygomycetes, Trichomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes were formed. During the last few decades, significant advances like recognition of the artificial nature of 3-kingdom and 5-kingdom classification systems, polyphyly of organisms traditionally known as fungi, these are accepted, and instead of the division, mycologists are gradually accepting the term phyla. So there is some change in the classification of the fungi. However, the important groups, classes of the fungi are Chytridiomycetes, Hypochytridiomycetes, Oomycetes, Plasmodiophoromycetes, Zygomycetes, Trichomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Phycomycetes is an obsolete taxon for certain fungi with aseptate hyphae. This class was earlier there. This class now has been abolished. This class has been abolished, and that is the organisms in phycomycetes are separated into chytridiomycetes, hypochytridiomycetes, plasmodiophoromycetes, zygomycetes, trichomycetes, and oomycetes. But earlier these are called phycomycetes, chytridiomycetes, hypochytridiomycetes, plasmodiophoromycetes, zygomycetes, and trichomycetes and oomycetes fungi together earlier called as phycomycetes. Members of phycomycetes are found in aquatic habitats and also on decaying wood, on moist and damp places as obligate parasites on plants, and these are most primitive fungi. These are most primitive fungi. Phycomycetes fungi are most primitive fungi because of their morphological similarity to algae. These are called algal fungi. Because of their morphological similarity, these are called algal fungi. Popularly, they are called as algal fungi because they bear a striking resemblance to some of the algae Vaucheria in morphology of the thallus and sex organs. So, why these are phycomycetes fungi are called algal? Why these are called phycomycetes and algal fungi? Because of their resemblance to algae such as Vaucheria in body organization and also in and similarity in sex organs. And these are regarded as degenerate algae by loss of chlorophyll and loss of ability to synthesize food, and they lost the chlorophyll and they lost the ability to synthesize the food and become fungi. These are some are aquatic, freshwater and marine, some are parasitic on animals, and some are parasitic on plants, and some are saprophytes on submerged vegetable and animal debris. Some are mycorrhizae, very few are mycorrhizae, and some are terrestrial, some are growing on decaying organic matter. The mycelium is unicellular or multicellular aseptate cenocytic hyphae. In phycomycetes, the mycelium is unicellular, or the mycelium is the body is unicellular, or it is filamentous, aseptate filamentous hyphae with cenocytic condition. Aquatic phycomycetes are commonly known as water molds. Some are amphibious in habitat, some are terrestrial in habitat. Some phycomycetes are terrestrial in habitat. Terrestrial phycomycetes are more destructive parasites of crop plants and certain insects. They cause diseases in some insects. And some phycomycetes, they cause spoilage of foodstuffs, spoilage of fruits and spoilage of vegetables. Amongst the phycomycetes, the most primitive ones have unicellular, uninucleate, uniflagellated holocarpic thalli with or without any surrounding membrane. So the most simple primitive phycomycetes, these are unicellular, containing single nucleus and single flagella, and the thallus is holocarpic, single cell. It acts as a vegetative unit and reproductive unit. In some advanced species, the somatic phase is a rhizomycelium, and in more advanced forms it is represented by a scanty mycelium consisting of a few short hyphal branches. The majority of phycomycetes have profusely branched, well-developed filamentous aseptate mycelial thallus. Most of them are eukarpic. Eukarpic thalli, in eukarpic thalli, at maturity, the thallus is differentiated into a vegetative part which absorbs the nutrients and a separate reproductive part. So vegetative and reproductive parts are separate in the thallus. Such thalli are called eukaryotic thalli. Whereas in most of the simple unicellular organisms, the single cell at maturity be converted into the reproductive organ. Such thallus is called holocarpic thallus. In phycomycetes, the hyphae or mycelium is aseptate. Aseptate mycelium is characteristic. Aseptate cenocytic mycelium is characteristic feature of phycomycetes. Multinucleated aseptate mycelium is characteristic feature of phycomycetes. The no septa in hyphae and mycelium. So there are many nuclei in the cytoplasm. Cenocytic aseptate hyphae or mycelium is characteristic feature of phycomycetes, lower fungi, algal fungi. During the development of reproductive structures, septum being formed at the base of each reproductive organ in some phycomycetes. The septa of phycomycetes are different from other fungi that they do not contain pore in the center. They are solid. The septa are solid. They do not contain perforation in the center. In phycomycetes, the mycelium is profusely developed where the reproductive bodies are less conspicuous. In the group as a whole, the development of fruiting bodies are comparable to those of other groups of fungi is absent. But in a few, there is a tendency of formation of fruiting bodies or reproductive structures. For example, endogone. In phycomycetes, there is no interval between plasmogamy and karyogamy, which occur almost simultaneously, but in most of them the post-sexual stages are not clearly known. In general, meiosis takes place before the development of new individuals. In small number of phycomycetes, for example, species of allomyces, however, meiosis of the zygotic nucleus is delayed, resulting in the development of a diploid structure which may be equally well developed like the haploid vegetative body, indicating the presence of alternation of generations. The life cycle pattern in the phycomycetes is rather simple in comparison with other higher fungi. It consists of a well differentiated asexual cycle and sexual cycle. The somatic body may be directly take part in the asexual reproduction or may produce sporangium from which spores are developed asexually. Each of these spore on germination produces somatic body hyphae. The somatic body depending on conditions may directly take part in the sexual reproduction or may produce gametangia from which gametes are developed and by gametic union zygote is formed. Again, the gametangia without producing well organized gametes may also take part directly in the sexual reproduction. As soon as the compatible nuclei are brought together by plasmogamy, almost immediately after that zygote is formed by karyogamy, resulting in the diploid phase. The diploid phase is followed by diplophase is followed by meiosis, leading to the development of spores or germ tube from which the somatic body is developed. The zygote may be thick-walled and become active only after a period of rest. Meiosis may thus be delayed. The major portion of the sexual cycle in the phycomycetes comprises of haplophase. Phycomycetes includes chytridiomycetes, plasmodiophoromycetes and zygomycetes. So these are earlier included under the Phycomycetes fungi. Class Chytridiomycetes. Class Chytridiomycetes is phylogenetically one of the oldest class in fungi. Phylogenetically, these are first formed fungi on the earth. Chytridiomycetes, commonly known as chytrids. Chytrids are saprotropes. Chytridiomycetes are chytrids. They have chitin cell walls and a posterior whiplash flagellum. Chytridiomycetes, They reproduce with formation of zoospores that are capable of active movement in aqueous phase in water. Chytridiomycetes reproduce with formation of zoospores that can actively move in water bodies, fresh water and marine water. Most members of chytridiomycetes, asexual reproduction occurs through the release of zoospores derived through mitosis. In some members, sexual reproduction is achieved through the fusion of isogametes. Chytridiomycetes are xenocytic with no distinction between individual cells. The filaments are long and tubular with a cytoplasm lining and large vacuole in the center. These single-celled organisms have branching hyphae with rhizoids. Chytridiomycetes are chytridiomycota. These are informally known as chytrids. The name is derived from the Greek chytridion, meaning little part, describing the structure containing unreleased zoospores. Chytrids means the name refers to the structure of fruiting body, little part which contains zoospores. Chytrid, the name refers to the structure of fruiting body, little part which contains zoospores. So the cell walls of these fungi contains chitin. The chytrids differ from other fungi in producing motile flagellated spores called zoospores, a possible remnant of their adaptation to aquatic environments, mostly freshwater and moist soils where they are commonly found. Chytrids are one of the early diverging fungal lineages, and their membership in kingdom fungi is demonstrated with chitin cell walls, a posterior whiplash flagellum, absorptive nutrition, use of glycogen as an energy storage compound, and synthesis of lysine by alpha amino adipic acid pathway. Chytrids are saprobic. They are saprophytes, degrading refractory materials such as chitin and keratin, and sometimes some are parasites. There has been a significant increase in the research of chytrids since the discovery of Batrachochytrium dendrobatides, the causal agent of chytridiomycosis in frogs. Class Chytridiomycetes has over 750 chytrid species distributed among 10 orders. The chytrids, they differ from many other fungi. Chytridiomycetes is the earliest branch of true fungi dating back to about 1 billion years ago. The chytrids, the cell walls are composed of chitin and glucans. They produce motile flagellated spores called zoospores, an adaptation to aquatic environments. Flagellated zoospore with a single posterior whiplash flagellum is characteristic feature of chytridiomycetes. They exist as single cells. Some chytrids form colonies with hyphae. Some are free-living forms that degrade organic material. Example, allomyces. Few chytrids are parasites of animals, plants and protists. Chytrids, these are simplest forms, these living endobiotically entirely within the hosts. Some species are epibiotic, producing their reproductive organs on surface of living host or on the dead and decaying organic matter and absorbing food by means of rhizoids. They contain rhizoids. Rhizoids are short, delicate filaments that contain protoplasm without nucleus and help to anchor the unicellular thallus to substratum. Some species produce an extensive system of branched rhizoids known as rhizomycelium. In more complex members, a true xenocytic mycelial thallus is present. In some complex forms, pseudosepta are present in hyphae. Example, allomyces. Chytridiomycetes are responsible for a decrease in the population of amphibians and salamanders. A variety of factors are involved in amphibian decline worldwide. Fungal chytridiomycosis is the condition. Fungal chytridiomycosis is the disease that is responsible for decline of amphibian population throughout the world. So the death of the amphibian species, mortality of the amphibian species that has resulted due to chytridiomycetes infection. So there is one chytrid, Batrachochytrium dendrobatitis. Batrachochytrium dendrobatitis, this chytrid causes chytridiomycosis of frogs, a condition in which chytrid infect frogs epidermis and interfere in respiration of the frog. Batrachochytrium dendrobatitis causes chytridiomycosis of frogs, a condition in which the organism infects frogs epidermis and interfere with the ability of the frog to respire across the skin. Chytrid fungi, these are responsible for massive die-off of amphibians worldwide, possibly in response to increase in the environmental temperature associated with global warming. So this global warming increase in the temperature, warmer temperatures improve the growth conditions of the chytrids. Thus, frog chytrid encounters are more likely to occur. These factors led to the mortality of frogs and amphibians and salamanders. Chytrid Batrachochytrium dendrobatitis causes chytridiomycosis in frogs and responsible for the decline of the frog population, amphibian population. Batrachochytrium dendrobatitis causes chytridiomycosis in frogs. Chytridiomycosis has resulted in the serious decline and extinction of 200 species of amphibians worldwide and poses the greatest threat to biodiversity. Chytridiomycosis is caused by Batrachochytrium dendrobatitis, and chytridiomycosis in salamanders is caused by Batrachochytrium salamandrivorans. Batrachochytrium salamandrivorans causes lethal skin infections in fire salamander, Salamandra, which has resulted in steep decline in salamander populations in northwestern Europe. Sexual reproduction in chytridiomycetes takes place by formation of zoospores which are born on sporangium. Sexual reproduction in chytrids takes place by planogametic copulation or by somatogamy. Oomycetes is another important class in fungi. The Oomycetes fungi are commonly known as water molds. These are a group of several hundred organisms that include some of the most devastating plant pathogens. The diseases they cause include seedling blights, damping off, root rots, foliar blights and downy mildews. Some notable diseases are late blight of potato, downy mildew of grapevine, sudden oak death and root and stem rot of soybean. So the oomycetes, these are causing number of diseases in plants. Oomycetes they have filamentous growth because of their filamentous growth habit and nutrition by absorption and reproduction via spores. Oomycetes were long regarded as lower fungi. Oomycetes were regarded by the plant pathologists as lower fungi. However, understanding of evolutionary relationships has grown. It is now clear that this group of organisms are unrelated to the true fungi. Unicellular or multicellular filamentous forms, they may be holocarpic or eucarpic. So unicellular and multicellular forms are there in Oomycetes. So multicellular forms may be eucarpic and unicellular forms, holocarpic forms are there. Oomycetes, they attack the higher plants They grow either intercellularly through dead and dying cells of host or intracellularly giving rise to haustoria. One of the most distinguishing characteristic in the oomycetes is production of zoospores produced in sporangia. The anterior flagellum of zoospore is a tinsel type while the posterior flagellum is a whiplash type. Both are typically attached in a ventral groove. So oomycetes possess two flagella. Oomycetes possess two flagella. One is tinsel flagellum, one is whiplash flagellum. So the anterior flagellum of zoospore is a tinsel type while the posteriorly directed flagellum is whiplash type. So this is characteristic feature of oomycetes. And zoospores, they retain a consistent and flexible shape although they lack cell wall. Zoospores can swim in wet water films and leaf surfaces, in soil water and hydroponic media and natural bodies of water. Oomycetes, the oomycetes fungi, they are characterized by production of these zoospores, and these zoospores, they swim in the water and they settle on the surface, retract their flagella and secrete a mucilaginous matrix which affixes them to the surface. Sporangia in different taxa within the group are of diverse shapes and characteristics. And in some species of oomycetes, zoospores has the ability to produce the zoospores has been lost, and the sporangia are thought to have evolved into the structures that directly germinate to produce the germ tubes. So such sporangia are called conidia. In other species, sporangia can germinate directly to produce germ tubes or indirectly to produce zoospores, a trait which is often temperature dependent, with zoospores being produced at cooler temperatures. Sexual reproduction occurs via the production of gametangia, oogonia, female, and antheridia, male gametangia. Sexual reproduction occurs by formation of gametangia, oogonia and antheridia. Because meiosis does not occur until the formation of gametangia, so the in the vegetative hyphae the nuclei are diploid. In oomycetes, antheridia and oogonia are formed. Each individual produces both antheridia and oogonia, and there may be sometimes a difference of plus and minus female and maleness. In some species, two distinct mating types occur, plus and minus. Both are required for the sexual reproduction. So these are called heterothallic. Two distinct mating types are there in some species, heterothalism. Heterothallic oomycetes, the gametangia are produced only in the presence of both mating types due to the fact that a hormone produced by one thallus stimulates the other to produce the gametangia. In other species, sexual reproduction occurs within a single individual, homothallic individuals. Unlike the heterothallic species, homothallic individuals do not require distinct mating types but can produce on same thallus. These are produced, gametangia are produced. All Pythium and Phytophthora species are homothallic. The fertilized oogonium develops into a thick-walled oospore. Oospores are produced, they may occupy a large portion in the plant tissues. The oospores many species have been shown to be able to survive for years in soil. Important oomycetes parasites are Phytophthora infestans. Phytophthora infestans, it is a parasitic fungi, plant pathogen. It causes late blight of potato, potato. This is caused by Phytophthora infestans. The great Irish potato famine was caused by Phytophthora infestans, which led to the death of 1 million Irish and emigration of another 2 million Ireland people in the Ireland during Great Irish Famine. Downy mildew of grapes is caused by Plasmopora viticola. another oomycetes organism, Plasmopora viticola, oomycetes fungi, responsible for causing downy mildew of grapes and causing heavy loss into the grape plantations. White rust of crucifers is caused by Albugo candida, oomycetes species. Albugo candida is responsible for causing white rust of crucifers. Glomeromycetes, glomeromycota or glomeromycetes include the economically important endomycorrhizae, arbuscular mycorrhizal fungi. Glomeromycetes fungi, earlier these organisms are included under the zygomycetes. Now they are separated from zygomycetes and placed in a separate class, glomeromycetes. Glomeromycetes or glomeromycota include the important fungi, endomycorrhizae fungi, arbuscular mycorrhizal fungi, vesicular arbuscular mycorrhizal fungi. The glomeromycetes are a relatively small group of fungi and they has ecological importance. There are very few species. There are only 230 species are there in glomeromycetes. Very few species are there in glomeromycetes. So these glomeromycetes are responsible for formation of mycorrhiza. They form vesicular arbuscular mycorrhizae in angiosperm plants. As many as more than 70% angiosperm plants form endomycorrhizal associations in which fungal hyphae produce swollen vesicles and arbuscules. So these fungi are called vesicular arbuscular fungi, where they form vesicles and arbuscules. These fungi grow in between the cortical cells in the root and produce large swollen vesicles. These vesicles store the food. They have cenocytic hyphae, multinucleate, aseptate, cenocytic hyphae are there in glomeromycetes. These hyphae penetrate into the cortical cells, few cortical cells, to form dichotomous tree-like branching structures called arbuscules. So these fungi, glomeromycetes fungi, produce vesicles and arbuscules in the cortical cells of the roots in angiosperm plants. The glomeromycota or glomeromycetes have cenocytic, occasionally septate mycelia, and they reproduce asexually through development of the hyphal tip to produce spores, glomerospores, with a diameter of 80 to 500 microns, and these complex spores they again germinate and form the hyphae. So glomerospores are produced by these fungi. These are not cultivated in culture media. In the laboratory, they cannot be grown in culture medium. Glomeromycetes fungi cannot be grown in culture medium. They grow only in plants. Without plant, they do not grow in inanimate media, culture media. 6 in ribosomal RNA sequence, in the ribosomal RNA sequence analysis of glomeromycetes shows that they differ from zygomycetes. Earlier these are classified in zygomycetes. Ribosomal RNA sequence analysis clearly shown the differences with zygomycetes. That's why they are removed from zygomycetes and classified as glomeromycetes. There are 230 species are there in glomeromycetes. Some important typical genera of glomeromycetes include Acalospora, Entropospora, Archaeospora, Glomus, Paraglomus, Gygaspora, Scutellospora. Zygomycetes is other important class in fungi. Zygomycetes are characterized by cenocytic hyphae and asexual reproduction by non-motile sporangiospores and sexual reproduction by gametangial copulation that results in the formation of zygospore within zygosporangium. In zygomycetes, cells are haploid. Cells have cell walls with chitin, chitosan and polyglucuronic acid. Cell walls contain chitin, chitosan and polyglucuronic acid. Zygomycetes are heterotrophic. They are saprophytes and parasites. They grow on decaying organic matter as saprophytes, rhizopus and mucor. And some are parasites causing diseases. And some zygomycetes are mycoparasites growing on other fungi. Some zygomycetes even grow on other zygomycetes as parasites. Zygomycetes have complex life cycles with both sexual and asexual reproductions. In the asexual phase, mycelia form sporangia. Within the sac-like structure sporangium, number of haploid spores are produced. At maturity, sporangium breaks and these sporangiospores are released. In favorable conditions, each sporangiospore germinate and form hyphae. Hyphae grow, ramify and form mycelium. Mycelium at maturity again forms sporangia. During the sexual reproduction, when hyphae of different mating types plus and minus hyphae come in close proximity, they form hyphal extensions called gametangia. These gametangia contain several nuclei. The fusion of two gametangia in gametangial copulation mixes the haploid nuclei together and form heterokaryotic zygosporangium. This zygosporangium remain dormant for some time. In favorable conditions, different haploid nuclei in zygosporangium fuse and become diploid nuclei in the zygosporangium. This is followed by meiosis and production of haploid nuclei, and each haploid nucleus is surrounded by bit of cytoplasm. Number of zygospores are produced inside the zygosporangium, and this zygosporangium bursts and release of number of zygospores takes place. These haploid zygospores in favorable conditions germinate and produce hyphae. Hyphae grow and form mycelium. Important genera of zygomycetes are mucor, rhizopus, phycomyces, thermomucor, syncephalastrum, pylobolus, thamnidium, piptocephalus. Thank you for listening the class on the diversity of fungi, biodiversity of fungi and different groups, classes of fungi.